Hello everyone, today we are going to review the structural mechanism of the Mre11-Rad50 complex for detecting and processing DNA ends. We will be examining the ideas of our article titled Mechanism of DNA End Sensing and Processing by the Mre11-Rad50 complex. In our article, structural states of an E. coli Mre11-Rad50 homolog, known as the ECMR complex, were studied to understand DNA end sensing and processing mechanisms. To review, the MRN complex is a complex consisting of the Mre11, Rad50, and MBS1 proteins. It plays a critical role in forming, processing, and sensing double-strand breaks in DNA, which are breaks in DNA that can induce cell mutations and be lethal to a cell. Prior research indicates that MR and MRN complexes can detect clean and blocked DNA ends, but the mechanism in which this occurs structurally is unknown. Additionally, although ATP is necessary for Mre11 nuclease activity, ATP-bound Rad50 blocks Mre11's nuclease active sites. Researchers aim to use cryo-EM techniques to resolve this data incompatibility and to propose how diverse ends are sensed and processed by ECMR. Rad50 has two coiled coils, also known as CCs, that can form ring and rod structures and are joined together by a zinc hook. Clean DNA ends refer to ends free of obstructing chemical modifications or structures, whereas blocked DNA ends are structurally obstructed by chemical modifications, including proteins and binding factors, and cannot be joined to other ends in a direct manner. EC Mre11 NUC refers to the nuclease active site of Mre11 in E. coli, and EC Rad50 NBD refers to the nucleotide binding domain of the Rad50 protein in E. coli. Endonuclease activity is DNA cleavage and resection within the DNA strand, whereas exonuclease activity is DNA cleavage and resection at DNA ends.
The researchers hypothesized that using cryo-EM to cell the full-length Mre11-Rad50 structure would show how the MR complex senses and processes diverse and blocked DNA ends. The key experimental techniques include cryo-electron microscopy and nuclease assays. Cryo-electron microscopy is an imaging technique for studying frozen biological samples where several 2D projections are summated to generate a 3D model of the molecule. Nuclease assays involve the use of radioactive labeling to visualize DNA substrates on an agarose gel.
Through cryo-EM and single-particle reconstruction, the structure of ECMR was determined to exist in two major states, the resting state and the cutting state. The resting state represents the state of ECMR in which DNA is absent and ATP is bound to Rad50. As seen in the labeled resting state structure, ATP gamma S was used in the absence of ATP to obtain a higher resolution structure. ATP gamma S is useful to obtain higher resolution structures of ATPases such as Rad50 because it is hydrolyzed very slowly in comparison to ATP. Because this substrate is so slowly hydrolyzed, it essentially sits in the active site, which allows for much more homogeneous particles to be observed during cryo-EM. Looking at the resting state structure, we can see that it is a homogeneous complex consisting of a Rad50 dimer and an Mre11 dimer. The Mre11 dimer is anchored to the Rad50 coiled coils via its C-terminal domains. We can see these two coiled coils coming off the Rad50 dimer, labeled CC. The entire structure of these coiled coils was unable to be obtained, but their angle of protrusion in this state suggests that they form wide, flexible rings. The presence of ATP engages the Rad50 nucleotide binding domains in this state, causing them to join together when binding ATP gamma S. This engagement of the Rad50 nucleotide binding domains effectively blocks the nuclease active sites on Mre11. Because ECMR is constantly exposed to ATP inside the cell but hydrolyzes it very slowly, this visualized ATP gamma S bound state likely represents a major auto-inhibited resting state of the ECMR complex. To determine how ECMR detects DNA ends, a nuclease-deficient ECMR mutant was used in the presence of both double-stranded DNA and ATP. The structure obtained from these conditions was deemed the cutting state, and the transition from the resting to the cutting state is currently being visualized on this slide. As can be seen, DNA end sensing induces two large conformational changes in the cutting state compared with the resting state. The first change is that the coiled coils move inward and zip up into a rod-like geometry and tightly clamp around a single double-stranded DNA duplex. The second major change is that the Mre11 dimer relocates from its auto-inhibited position at the bottom of the Rad50 dimer to the side of the Rad50 dimer. Here, Mre11 and the Rad50 nucleotide binding domain form an active site channel that harbors the DNA end. Another observation is that only one of the two Mre11 subunits contacts the DNA end in this state. The subunit in contact with DNA is referred to as subunit A, while the other subunit helps to position subunit A and is referred to as subunit B.
To form the cutting state, the phosphoesterase domain of Mre11 is connected to Mre11-Rad50 by a loop, denoted as fastener. The fastener binds the outer beta sheets of Mre11-Rad50 in yellow and holds both domains in place using polar and ionic interactions. To validate the interface, the central K149 and E115 salt bridge connecting the fastener and Rad50 were mutated in DNA1 and DNA2 to show endo and exonuclease activities. DNA1 had only a fluorescent green label, and DNA2 had both a label and a protein block. In DNA2, the blocked DNA revealed higher endonuclease activity than DNA1 because DNA with free ends do not need as much endonuclease activity. Individual mutations reduce the activity of both endo and exonuclease activity. Mutating the charge of both amino acids restores exonuclease activity in DNA1 and endonuclease activity in DNA2. This is because originally K149 is positively charged and E115 is negatively charged. Changing them both allows salt bridge interactions. Overall, these results validates the cryo-EM structure such that the endonuclease activity in the cutting state occurs only in blocked DNA.
There is a nuclease active site, the location for Mre11 nuclease activity, located in a transient channel between the Mre11 and Rad50 proteins, as shown in the image. Mre11 subunit A holds a DNA end to cut the strand. The DNA end is held in a dimanganese cluster within the nuclease active site to be processed. The transient channel can explain the MR complex's broad specificity for different DNA ends, as the channel can accommodate various DNA structures.
The coiled coils fold into a rod structure and bind to DNA. Regarding the shape of the CC rod structure, the folded interruptions are to form the zipper contacts and create the rest of the rod structure. The zinc-cooked diamonds which join the ends of the CCs act as a hinge which allows the CCs to open into a ring or close into a rod. The GERR motif is a highly conserved sequence among the bacteria MR homologs that can mark an important site near the first CC zipper contact. An assay was ran where the GERR motif was mutated to AEIR. The results show that the GTA mutation significantly reduced endonuclease activity on free and fluorescent blocked DNA ends. However, the exonuclease activity is about the same as the wild type. This shows that the change to G to A is critical for ATP-dependent cleavage of blocked DNA.
Endonuclease specificity towards certain DNA structures is suggested by the preferential entrapment of linear rather than looped DNA structures. This was tested by observing cleavage patterns of a linear DNA fragment blocked by a streptavidin molecule at each end and a circular DNA structure blocked at both ends with one streptavidin. It was found that the addition of ECMR led to the degradation of the blocked linear DNA structure, represented by Roman numeral 2, and not the circular structure, represented by Roman numeral 3. The ECMR's binding affinity for linear DNA was also found to be higher than that of supercoiled and relaxed circular DNA, regardless of whether the DNA end was clean or blocked. Thus, the discriminatory feature for DNA end recognition and processing is likely the topology of DNA. It is suggested that the CCs play a role in DNA end detection by employing a gating mechanism, forming rods when a block on a single duplex rather than a loop is detected. Overall, in order for DNA end recognition and processing to occur, a DNA block must reside on linear DNA.
When loading onto blocked DNA, the Mre11 dimer needs to relocate from its normal conformation to climb over the protein block to the DNA ends. For this to occur, Mre11 would have to open up, so the researchers looked at whether dimer dynamics had a role in this relocation. They fused a LISH dimerization domain with Mre11, which essentially acts as a tether between the two Mre11 domains. Next, they assayed degradation of the linker length between Mre11 and LISH with 11 and 20 residues. Here we see that LISH11 has little endonuclease activity, while LISH20 shows delayed endonuclease activity. This might be because LISH20 allows Mre11 dimers to disengage and re-engage on the other side of the DNA. This suggests that Mre11 dimer has to be dynamic, particularly for endonuclease activity.
To conclude our presentation, the results here revealed that there were two different states of the ECMR complex, the resting state and the cutting state, and the resting state represents the major autoinhibited state of the ECMR complex. And so the cutting state is triggered by DNA binding, where the Mre11 dimer moves to the side of Rad50 to form a DNA cutting channel, and the CCs form a rod-shaped DNA gate and clamp. And so the analysis of the fastener loops revealed that this stretch of amino acids is critical for initiating the cutting state structure through salt bridge interactions between the phosphoesterase domain of ECMR11 and EC-Rad50. It was also discovered that the endonuclease activity only occurs in blocked DNA, and this is likely because the DNA with free ends can be excised with exonuclease activity alone. And so when loading linear DNA, the DNA block must reside on linear DNA for recognition and cleavage, and CCs employ a gating mechanism that includes rod formation initiation, when the single duplex blocks are detected. And so when linking the Mre11 dimer, it was found that the Mre11 dimer relocates from its normal conformation, and researchers suggested that the dimer had to be dynamic to facilitate movement, particularly for endonuclease activity.
Moving to further research, one question that arose from our paper was what is the exact shape and sequence of the Rad50 coiled coils? The resolution of the methods used in this study were not sufficient enough to resolve the entire coiled coil structure, but future studies with better resolution could resolve the entire structure, giving us better insight into the role that the coiled coils play in the MRN complex. A second question that arose from our paper was what is the precise orientation of ECMR during the endonucleolytic processing of blocked DNA? While current data supports a model in which the DNA blocks are on the Mre11 distal side of the complex, further research, specifically a structure of the ECMR complex bound to blocked DNA, is needed to confirm this speculation.